Hi everyone. So my name is Mohit Gupta. We all know each other. We have been member of our family since a long period. Today, why am I standing over here? The main question is that I am standing over here with a new topic that I am going to teach you. One of the topics that is most interesting one, and one of the topics that are to be taught very there is a difficulty to understand these topics for the students. So we are going to help you to recognize, to memorize these topics. So let's go. So what is today's topic is the central dogma of life. We all know that this topic is very precious topic because this topic is underwent from million years, right? Since the era of evolution, if the central dogma would not have been working over there, then it might be the possibility that a person would not have been evolved. This is the best possibility, that the humans have not been evolved, or any creature would have been not evolved. There will be, or there must be a single species all over the global area, or we talk about the global type of categorization. There will be only one single species if this central dogma would have been not worked, right? So we have been talking about this central dogma in detail. I have talked about earlier also about this central dogma, but the processes, the propagandas are not that much clear that time. Now let's talk clearly about the central dogma. Now the main question arises, will I be only talking about it in a single video? No, this is a vast topic and we need certain time limit to talk about such a vast topic, and time limit is somewhat maybe merely a week, maybe merely a month. So any time I can take, because it is a very vast topic, right? And we need certain time to elucidate such a vast topic, right? Now let's start. Very firstly, this is the introduction video, guys. I will make certain playlist for this video. I will choose a particular name, that is the Central Dogma of Life by Mohit Agriculture Tutorials, present to you all, guys, because it has been one of the bestest lecture ever on our channel, because this is a very vast topic. Now let's talk about precisely what is Central Dogma of Life and how in vivo it works in our body, how we can recognize this whole criteria in vitro. I'll talk about this also, that how scientists recognize it, but in vivo it has always been going on, even though we are familiar with the agriculture, right? So in agricultural criteria of studying, plants are also being evolved into different categories due to the same type of working, right? Same type of working, that is central dogma of life. If central dogma of life would have been not anywhere, then we should not see such a variation among the species in global age. Now let's talk about what is central dogma. Look, central dogma is very we can say simple topic. We all have heard about it in our intermediates, high schools, even though in graduation. If you are a graduate from science stream, then you have been heard. If you're not graduate, then no problem. We'll let you recognize, let you memorize the topic, that is central dogma of life. Let's start. Central dogma of life. It is the topic that starts with single thing, that is DNA, or we can say a gene. What is a gene? Gene is somewhat mixtures of four base pairs: adenine, guanine, cytosine and thymine. These all bases, they merge with each other, and when they merge with each other, certain phosphate bonds and all those things, phosphate backbone and all those things, when hydroxyl bonds and all those peptidyl bonds, and when they merge with each other and they form a structure, that is a double helical structure known as DNA. We'll talk about it in brief when we are talking about replication, transcription, translation. But for now, just understand the overview of the whole topic, because the whole topic is very, very vast. And sorry for my language, I have introduced a term that should be not in this topic, that is peptide bond. That should be in this topic. Topic is central dogma. Peptide bonds are not in DNA. The bonds, peptide bonds, are solely in the proteins. That's why we call it polypeptide chains. Now, why I have introduced this word to you? There is my particular intention for introducing such a word, right? Because central dogma of life is working merely on the basis of protein foldings, protein primary, secondary, tertiary, quaternary structure, alpha, beta sheets. All those when merged with each other, we know about the process of the central dogma in detail. Now, let's go through what is the central dogma. Let's see. Okay, so here I am ready. So the process starts with a single word, we can say a 3-letter word, DNA. DNA is the king of the nucleus in a cell, right? When it is in the nucleus, various enzymes are being working as a, you can say, drone, or working as workers over there. We may think of a apis culture, bee culture. We are being what say we are adding certain bees in our, say, a plantation, or say a tree. We are adding certain bee and we are just culturing them somewhere, right? Now, during culturing them, we see lots of things that are being inspiring this topic, that are very, very much related by this topic. Bees are being living in their, we can say, shed, like a queen, right? Like a queen, the bee that is being fertilized furthermore by the drones, and workers are being serving her for laying the eggs, right? So somewhat we can think like that type of propaganda, that type of process going on in the DNA, in the nucleus, sorry, in the nucleus. DNA is the queen of the, we can say, this is the shed where the bees are living, right? Sorry, I am not getting the particular word that I should use for the bee. Where do they live? I am not getting the particular word. Again, let's say it a shed, right, where the bee lives. Now, particularly this is the shed, and inside it the DNA is has been living over there like a queen. Certain enzymes are there. Enzymes are working as a drone. They are serving it. They are reproducing it. They are trying to fertilize it, and they are somewhat say replicating it. Right? Replication is the correct word. So DNA is being replicated inside the nucleus. It is the king of the nucleus. Once DNA is replicated, now the process goes on. DNA to mRNA, mRNA to tRNA, and tRNA to protein. And this process that we are talking about in series 1, 2, 3, 4, and so on. The process goes on. Every time once protein has been built, the many more process going on in the cell. Cell organelles are working very, very efficiently for the protein processing. So we'll consider that in detail later. Now let's talk about this process. So the overview of this process is very simple. Now let's, to understand this topic, let me have a good idea. The idea is that we are considering certain traits over here. Right? So now you can understand that what is the topic by considering certain traits over here. Say, traits, what are the traits? Go back to my genetics lecture. Over there I have introduced the term trait over there. Trait, or we can say phenotype, genotype. We are using somewhat phenotype information, phenotypic information of individuals over there. Which individual we should consider? We should consider filmmakers, we should consider politicians. That will be a big drama type of sequence that would be created if I have used their name. So the name will be very simple, that I am being using over here, that is X, Y, and Z. Guys, sorry for the fault in between. I have went because I make myself my videos. There is no team with me, no person with me over here. I just personally make this video via my mobile phone. So some we can say problems are coming continuously in our shooting procedure. So don't try to look at in a negative manner, but always think in positive manner. I am giving you a biggest topic that I can introduce to you, but so think it like that. Okay, so we are considering three traits over here, three persons over here. Say person one is X, Y and Z. Now what is trait in them? What are the phenotypic information they contain? They contain height, perspective, height, say face, how are they looking by a face, what are their features of the face. Now we are considering two traits over here, right? And two traits of Y also, that height and face, height and face. Now these three traits, height, face, face, they are to be recognized of the three individuals that must have certain variations in them. The variation, now how can we recognize? Now how I will be labeling the variation? Say this person is tall, so H. This person is little dwarf, so H dash. This person is somewhat more dwarf, and we can say that this person is variation. That variation he is having in his genome. The genome is coding for some proteins that are being working in his body language in somewhat manner. That is phenotypic criteria of height is somewhat stunted height he is having. So we can consider this all things like that. That this one is tall, one bar, so H. Somewhat dwarf, not merely dwarf. It is approx height that a person should have 5. This is 6 feet, and this we say 5'5", so that's not bad. That's appropriate, okay, but not that much, but appropriate height. This person is having, Y is having, and this person is having dwarfness, a allele that is coding for dwarfness. Now this thing you should remember the allele and what are the traits. I have already mentioned these words in my previous videos of genetics and plant breedings and all those things. So don't merge it with any another word, right? Let me firstly try to express myself, then I'll be forthcoming to these words also, and I'll discuss what these words specify, right? So this H is, we can say, complete tall, this is height deficient, and this is dwarf, right? Now these are the words, correct? Not allele for the face, somewhat round face, FR, somewhat we can say oval face, so somewhat egg type of face, and this face is somewhat square face. So now here comes the central dogma. Why I am introducing so much traits, so many varieties of persons to you. If you can see from H to H bar, is it a mutation? What do you think? This is a mutation? No, this is not a mutation. If mutation would be there, a thing I should clear over here is we all are mutants. We are all are having central dogma somewhat mutated. That's why we are seeing the variations. But we don't call these mutations as mutation because these are not bad for us. If variation, somewhat variation, it is not bad unless it is somewhat hurting us or somewhat getting our body into a disease. So we'll not consider any trait. We are not considering any trait in these two cases, right? These are variation because their genome effect do not produce any threat to their individuality. But when we talk about the Z case is say square shape, abnormal shape, and height is merely abnormal. So this person might be in a threat. maybe his genome may be having some more malfunctions that may produce certain circumstances when this person might die, because there are already two mutations. It may fundamentally, when we'll what we'll do, that we will when extract the genome and then we'll read the information from this person's genome, then we'll know that the Z category function must have some more mutation that are hidden inside. Now, what is the difference between mutation and variation? Variation is somewhat plus, right? It is not minus, negative. It does not have any negative effects. It is just we can say protein coding and somewhat folding, misfolding, and so it's quite okay, right? But this protein folding and misfolding is producing an individual that is being in the thread. This is the process of central dogma. Central dogma is producing somewhat individual that are in the thread, or it may be positive also. Central dogma is working for our body regarding whatever functions our body is doing. Merely we are sleeping, we are walking, we are fighting, anything we are doing, the process is made very simple. Central dogma working over there. But when central dogma is somewhat threat to anyone, then we say that the dogma is in threat, or we say dogma is in negative coordinations, right? So these are all the propagandas of central dogma of the life. We'll talk little more detail in this video, then I'll go in further vast detail of this topic in merely many videos. I'll continue this series. So this is what is central dogma of life working like, right? So okay, now what else I have to discuss over here? Okay, the discussion is the process limited over here only? Means protein, once protein is coded, is this process not to be fundamentally say so on and so on continued? It will be continued. Who will continue this process? The process will be continued by now say certain organelles. Endoplasmic reticulum coming in RNA to protein process. Here only endoplasmic reticulum is coming. Then from here to Golgi apparatus. Golgi apparatus too. We can say plasma membrane, and many more functional organelles are coming in between of this central dogma. So these all are the categories of coordination in the cell. The cell is coordinating certain processes inside its own, right? So let's consider this whole gigantic process to be a single legal mass. We can say this all gigantic, this gigantic body is working like a coordination panel inside our body. If it is not inside our body, then this would have been of no use, because this is only useful for the living beings. That's why DNA, this is the master of the cell, is in the very first stage. Now, how it works, let's talk about this sequence, then we'll end this video and we'll continue this topic in next series. So I'll say that watch the whole series if you want to participate in competitive examination, because this is going to help you a bit, or we can say a wide helping nature of these video will be for you in some competitive examination. Let's see what else I have to talk in this video. Okay, let's go. Okay, so let's see what processes we will be talking in this. Very first process will be replication, DNA replication, transcription, translation. After that, RNA splicing, modifications after modification we'll go to somewhat genetics, and in a bit modification will be in two processes. First will be DNA modification, second will be cellular modification. In cellular modification we will be going to talk about somewhat what cell does in the modifying any protein in modifying, how cell organelles work together in a unity, how they transport protein from the one cell to the another, how protein works in the cell, what what are the after the protein coding sequences, what are the modifications that protein is gaining beside this process. So these all things we will be talking in this series of lectures, right? So hope so that you will enjoy this series of lectures, and this series of lecture will be in complete English. I want you to have good understanding of English, since my English is weak. So I want my English also to be improved in this series, and at the same time your English should also somewhat improve it. If it is already improved, then don't discourage me by writing any comment in the comment section, right? I am trying to learn English, that's why I'm making English videos. This whole series will be in English. So okay, thank you, and what I say at the end, Jai Hind, Jai Bharat. Thank you.